Welcome, everyone, to what is our last event of 2024. I just wanted to thank all of you for your participation in these marvelous talks. Your enthusiasm and interest for learning are truly remarkable. And by this, you make our work here in Abu Dhabi quite meaningful. I also like to thank the staff here who's setting up all these talks. So thank you, everyone. I wanted to mention that although this is our last talk of 2024, we kick off January 2025 with a big bang. This January, the NYU Abu Dhabi Institute invites you to public talks exploring the frontiers of science, culture, and global challenges. In fact, we're calling it a month of science and exploration. I'll mention a couple of the upcoming talks so you can mark it on your calendar when you come back fresh for the new year. On January 8th, Stephen Schenker of Stanford University will be speaking about chaos, black holes, and quantum mechanics. On the very next day, January 9th, David Tong from the University of Cambridge takes us on a journey to uncover the origins of the universe. On January 13th, Jim Gates, who's a winner of the National Medal of Science in the United States and a professor at the University of Maryland, he's going to give a talk bridging physics, mathematics, and art. And the talk is entitled "The Islamic Tradition of Art and Mathematics, Quantum Gravity, and Our Understanding of the Universe." On January 22nd, we turn from all of that wonderful science to another thing that is a passionate interest here in Abu Dhabi, even in our fields, which is cricket, and where you can come explore the intersection of sport, politics, and identity with Ali Khan, who's an author of an important work on cricket. And finally, on January 27th, you can join us for an important talk on International Court of Justice, I'd say. So can justice be achieved in the devastation of modern wars, with a leading jurist. So there's a lot going on. So mark your calendars. Please follow us, head out to the websites, check us on social media, etc. And do have a very happy new year. For now, it is my pleasure to introduce my colleague, Dr. Alan Healy, who's an assistant professor of chemistry at NYU Abu Dhabi. Dr. Healy received his undergraduate degree in medicinal chemistry from Trinity College Dublin in Ireland, followed by a master's in science from the University of Edinburgh. And he obtained his PhD in chemistry in 2014 at the University of St. Andrews. And I guess relevant to this talk, at Yale University, he was part of a team that studied the link between small molecule toxins produced by select strains of bacteria in the human gut and the development of colorectal cancer. So with that, and more broadly, I should say, Dr. Healy's research at NYU Abu Dhabi is focused on using genomics, bioinformatics, and automated chemical synthesis to discover, study, and repurpose natural products as novel therapeutics. So with that, I give the floor to Dr. Alan Healy.
Okay, thank you for that lovely introduction. And I'm now going to take over the introductions. So good evening, ladies and gentlemen. It's my distinct pleasure to introduce tonight's Institute speaker, Professor Paul Hergenrother. So Paul was born and raised in Akron, Ohio, and he started his academic journey in the University of Notre Dame, where he obtained a Bachelor of Science in Chemistry. He subsequently obtained his PhD in the University of Texas at Austin. Following his PhD, he joined the lab of Strutschreiber in Harvard, and as an American Cancer Institute Fellow. And it was around this time that the Schreiber Lab was really at the forefront of translational drug discovery in academia. And Paul contributed to this by the development of high-throughput screening assays. In 2001, Paul joined the faculty at the University of Illinois Urbana-Champaign, where he quickly climbed through the ranks and was made full professor in 2010, followed by being appointed as the Kenneth Rinehart Endowed Chair in Natural Products Chemistry in 2013. And Professor Hergenrother's lab has contributed hugely to advances in basic chemistry, medicinal chemistry, and biological chemistry. But he's also incredibly well known for the translational aspects of his lab. And so he has had very promising anti-cancer compounds from his lab have been licensed by Bayer, Oncotech, many very big pharmaceutical companies, but also he's developing them through clinical trials himself as part of Vanquish Oncology, which is a startup that he co-founded. Beyond cancer, during the COVID pandemic, the Hergenrother lab developed a very simple, cost-effective saliva-based assay to, as a test for SARS-CoV-2. This was hugely successful and was used by the University of Illinois as their main screening method, with over 1 million tests being done using this assay. And it got priority emergency authorization and was used by many other universities, schools, and businesses during the pandemic. So that kind of translational research has obviously led Paul to be awarded numerous accolades for his research. Far too many to go through here, but I will just give you a brief overview or kind of synopsis of some of them, such as the NSF Career Award, the Beckman Young Investigator Award, the Alfred P. Sloan Foundation Fellowship, the Camille Dreyfus Teacher Scholar Award, ACS Eli Lilly Award, the UCB Ehrlich Award for Excellence in Medicinal Chemistry, National Cancer Institute Outstanding Investigator Award, and quite recently he was made a Fellow of the National Academy Inventors, which really highlights his role as a leader in translating scientific discoveries into real-world applications. And so today, I believe Professor Hergenrother is going to talk to us about another very critical area of research in his laboratory, which is the challenge posed by antibiotic resistance and the dwindling pipeline of new antibiotics. This issue is an imminent health crisis. Those of my students who are here have heard about it in my class. And this is something that will affect everyone in this audience unless we address it with urgency. And so with that, I would like you to join me in welcoming Professor Paul Hergenrother to the stage.
Great. Thanks, Alan. Thanks so much for that wonderful introduction, and thank you to the Institute for having me. I've had a great few days here in Abu Dhabi. I'm excited to speak with you all tonight, and thanks for coming to this lecture. So I've titled this Rethinking Antibiotics, and as mentioned, there is a looming crisis in drug-resistant bacteria. But I think the other thing that's just now coming to the fore that I'll talk about tonight is just what these antibiotics are doing to us. And that's just been a revolution in the last 10 years or so with modern DNA sequencing technology. And now we can really see how the devastation it does to our good bacteria. And just thinking about how, if we could kind of scrape off the drug discovery process and do it differently, how we might do it. And so that's what I hope to convey to you tonight. So I just want to orient you all with antibiotics and how we got from where we've been to where we are now. So there's this famous discovery, right? One of the most famous examples of serendipity in science by Alexander Fleming 100 years ago or so now, where this fungus contaminated this agar plate that had staph bacteria on it. So this is Staphylococcus gram-positive bacteria, and you can see this zone of inhibition here. So this suggested that the fungus was making something that killed the bacteria, and that after a lot of work, and many of the best chemistry minds of the day were involved in this 10-year process of figuring out what the structure of this was, which was very difficult back then with the technology available to them. Ultimately, that activity was attributed to penicillin, this compound shown here. Penicillin, of course, has had an incredible impact, and it really was the dawn of the modern age of antibiotics. attributed with winning the war for the Allies. You can see this propaganda from the day. And the ability to mass produce it was a whole nother amazing story and of science and engineering. And I think the most telling stat is here. So before penicillin, the lifespan was 54, and now still is something around 79. And this is because this antibiotic era facilitates surgery. We couldn't have surgery without antibiotics, not to mention routine infections that one used to get that could also often be fatal. And you hear stories of children 100 years ago or more that get an infection from out playing and then they succumb to it. So really a revolution. This led to what is called the golden age of antibacterial discovery in the 20 years subsequent to World War II. And the Waxman method pioneered by this gentleman. Really the observation was like, can we, this serendipity that led to penicillin, can we make this happen on a more routine basis, right? So you pick up a handful of dirt and there's millions of microorganisms in there, and what do they produce, right? And so at that time, it's like picking the low-hanging fruit from a tree. So the major classes of antibiotics that we use today were discovered then, aminoglycosides and macrolides and things like vancomycin, and were discovered in this era, and then over the 20 years or so after that were developed into drugs. And there was actually a thought towards the end of this that maybe the crisis of infectious diseases was over, right? Like we had solved the problem. Of course, that's not true, and the reason is this drug resistance. Just like you can hold millions of organisms in the soil, bacteria have the numbers advantage on us. So a flask of growing bacteria contains billions of bacteria. And so even though a mutation might be a rare event, and a mutation that causes resistance to a drug is even more rare, when you have those kind of numbers, resistance is inevitable. And so this is shown here. So in this graphic, here is the FDA approval year, and then on the bottom is when resistance is observed. Okay, and you can see the twinning of these two for different classes of antibiotics, and this is true for the initial discovery of a new class, and then follow-on drugs, same members of that class, and on and on. This is not the comprehensive list, but the same story has played out over and over and over with every antibiotic, and it's probably the way it's going to be, right? And so this is why one needs a continuous supply, and it's different than a disease like cancer, where you might expect to be able to cure it and solve those problems. Whereas here, bacteria have this advantage, so there needs to be a continual cycle of drug discovery. Okay, so bacteria can be broadly classified into two major categories, some called gram-positive and gram-negative, and these are classified based on their outer membrane. So gram-positive bacteria lack this outer membrane that gram-negative bacteria have. Okay, so gram-negative bacteria are intrinsically resistant because of this outer membrane. So this outer membrane makes them incredibly impenetrant. So would-be antibiotics that might be active oftentimes can't even get in. And if they get in, they're subject to these pumps, efflux pumps, that pump them back out. Okay, and so this has led to this situation where we have now, where there's a growing number of gram-negative and drug-resistant gram-negative infections because of this intrinsic resistant problem. And that's shown here. So in this slide, the observation has been there's no new antibiotic class FDA approved for gram-negative infections in 55 years. And so given the numbers advantage that bacteria have on us, this has led to rampant drug resistance. Okay, so this is deaths, and this is cases. And so MRSA is an infection many have heard of, and if you work in a hospital setting, you know about this. This for a long time was the most prevalent by far, but that's been overtaken by E. coli. These red negatives are all gram-negative bacteria. So you can see these 5 of the top 6 or 6 of the top 7 are drug-resistant gram-negative bacteria. And this paper shows how the arc of this is like rising exponentially. And there's some real gloom and doom numbers for this. But at this point, gram-negative bacteria are responsible for about 3/4 of the deaths that we see. Okay, and so the other major point that I want to make before going into what we've been doing is this, and I mentioned this at the beginning. So these antibiotics that we have, we've developed them to kill bacteria, and to kill bacteria, not to kill human cells, which is a big challenge. Okay, but bacteria, in some, by some counts, as people, we have more bacteria than we do human cells, okay? And we have a lot of good bacteria that help us with all sorts of things, from digestion to defense, to breaking down food, etc. And so the antibiotics that we have, we'll have a gut flora, so bacteria in the gut, a microbiome, and we've all taken antibiotics, and now you're killing the pathogenic bacteria, sure, and that's the goal, right? And that's been the priority. But now with DNA sequencing, so there's been a revolution in DNA sequencing in the last 15 years, and one can collect fecal samples and look at the composition of the gut microbiome, and it's been eye-opening. The devastation that happens to your gut microbiome when we take antibiotics, like basically wiping it out. As you can imagine, right? These are drugs, powerful drugs that kill all of these bacteria that are very closely evolutionarily related. So the numbers are down, the diversity is down, and it's just like every month there's a new discovery about what that's doing to us. So even 2 years later after taking this, we now know that our microbiome haven't recovered. There's all sorts of ways you can think about trying to reintroduce a microbiome, but a lot of those are just not effective. And so wouldn't it be great if we could develop a drug that just killed the pathogen, right, and didn't kill the commensal bacteria? So I think the major one is Clostridium difficile infection. And again, if you work at a hospital setting, you know this is a major problem. People where you wipe out the gut microflora, now this opportunistic pathogen, something that would not be pathogenic normally, but because it has no competitors, it takes over. Very, very hard to treat. But also now, no doubt, irritable bowel disease. If you live in the US, where we have commercials for drugs, you're constantly bombarded for commercials for Crohn's and colitis, with drugs for that, and there's just a huge industry trying to deal with the secondary effects from this, caused by this gut dysbiosis, colorectal cancer, as was mentioned in the introduction, but even things like now depression, exercise motivation, diabetes, immune dysfunction, etc., seem to be linked somehow to gut microbiome disruption. Okay, so this is like the appreciation of this problem has now been growing. Okay, so this leads me to the title of my talk, Rethinking Antibiotics. And so what I'll talk to you about today, our work, is really thinking about a new strategy of discovering antibiotics. It's been 55 years since a new class has been approved. I'll just show you a little bit about how it's been futile to try to do things the old way. It's not been successful. And then these new antibiotics, we want to be selective for pathogens over commensal bacteria. Again, a big challenge given just how tightly related pathogenic bacteria are to gut commensals. So I'll tell you about our discovery of this compound that we call fabomycin. That was discovered through this process that I'll outline, and we just have tiptoed with fabomycin into this selectivity, and then really have gone all the way into it with this compound, lolamycin. Both of these compounds we've recently licensed and are advancing, and hopefully will make it as antibiotics. But right now, I'll just talk to you about the strategy and the discovery. Okay, so the challenges of gram-negative antibacterial discovery, and I give kudos to our friends in industry for publishing what has been negative results, which oftentimes don't see the light of day. Okay, but GSK published this very sobering paper that I think a lot of us in the field read. 3 million compounds they tested, and they got 0 actives for gram-negative bacteria. AstraZeneca, similar, 2 million compounds, a bunch of screens, a few hits, 0 have gram-negative activity. Merck, 3 million compounds, 0 with gram-negative activity. And so this is like 0 for 8 million combined, and these are by experts, people that are really, really good at this, teams of hundreds of people spending a long time doing this. And I think the good part of this is that from this, we've discovered a lot of compounds that are active against gram-positive bacteria, okay? As I mentioned at the beginning, they don't have this cell wall, and so it, in many ways, are easier. And there's a whole interesting cornucopia of compounds, some of which I show here. There's at least 70 of these compounds. All of these, the interesting thing is, all would be active against gram-negatives if they could get in. Okay, and we know this from, you can artificially make gram-negative permeable, and you see that these compounds can kill. So I'll just show the example of this compound here. This is a really interesting molecule, fatty acid biosynthesis inhibitor, okay, shown here. This compound, very potent against staph. So the way that in the field we do this is called the MIC assay, minimum inhibitory concentration. So the lower the number, the better. So 0.002 micrograms per mil. So very small quantities of this are required to kill staph. Really has no effect against gram-negative bacteria, E. coli. But if we take that same E. coli and make it deficient in some way, not able to pump the drug out or permeable, the compound will now kill, okay? So we knew that from this and other work that these antibiotics that we have would have tremendous potential against gram-negative if they could only get in, if they could only get into the bacteria and stay in. Okay, so this led us to our first goal and our experiment where we wanted to totally divorce antibacterial activity from compound accumulation within the bacteria and not worry so much at the beginning with killing the bacteria and just ask the question like, what does a compound need to have to get into the bacteria and stay in? Okay, and just really try to study and understand drug accumulation in bacteria. Once we have a good understanding of that, then we can hopefully go back and build antibiotics with those features. Okay, so this is the workflow where we have a collection of compounds. I'll just talk briefly about this. And then we do what is a very painstaking assay assessment, one at a time of these compounds for their ability to get in and stay into bacteria. We use artificial intelligence and machine learning, so just looking at chemical traits of the compounds that accumulate and those that do not, and try to recognize traits that enable the drugs to get into the bacteria. And then, only then, after we have this, do we use this to develop new antibiotics. Okay, the compound collection is a whole story. I gave a lecture in chemistry last Thursday and talked about this. Just briefly, that 0 for 8 million taught us that the compounds in large screening libraries, industry-style screening libraries, are probably not the ones to use, right? 0 out of 8 million. These compounds tend to be, from a chemistry perspective, simpler molecules, tend to be flat molecules, and there's certain features that they don't have. Many antibiotics are from nature. They're isolated from soil, as I mentioned, and they tend to be complicated, more complex molecules, again, as measured by certain traits from chemistry. They tend to have contiguous and overlapping ring systems like you have here. And they have certain functional groups that are just present that are not present in synthetic compounds. The challenge is, you can get 8 million of these compounds because they're easy to make, okay? And you can't isolate. There aren't 8 million natural products that we can isolate readily. So before we did this whole, went into this whole workflow, we first needed to develop a means where we could get access to lots, hundreds, 1,000 complex molecules. Okay, and this is, we call this the complexity to diversity strategy. And my postdoc at the time, Rob Higgins, was really the first one to do this in my lab. He's now a professor at the University of Georgia. And what we do here is we take a natural product, so we can buy 100 grams of it, say, and then just using strategic application of organic chemistry, we change these molecules into things that are different from each other, different from the parent, but still complex compounds. Okay, and so we're using these natural products as a starting point for compound generation. There's a lot of readily available natural products, so from these various organisms, These are all interesting and complex molecules, and so we buy these, and then in the lab we change them into things that look different that are still complex molecules. Okay, and so like I said, I went into the chemistry of this the other day. I won't do that here, but we've made 1,200 of these compounds, and this is like it's a tangible resource, right? We have 10 milligrams of each in a vial. Okay, and you can see a plate here. It's a vial, and we have a bunch of these. Okay, so now we are ready for our big experiment, and I give a lot of credit to my former student, Michelle Richter, for taking this project on. Andrew Riley, Michelle's now at Vertex, Andrew's now at UI Chicago as a professor, for applying these rules first. So what Michelle did was took this collection of compounds and again measured one at a time their ability to accumulate. Okay, and so then we had accumulators, non-accumulators, and we looked at 300 different properties of those accumulators and non-accumulators, and developed what we call the entry rules. So these are traits that the accumulators have in common. And the entry rules stipulate that if a compound has an ionizable nitrogen, a certain three-dimensionality, so it's more flat versus globular, and a certain rigidity, it has a good chance of accumulating. And so we can just do tests. So this compound has an ionizable nitrogen. These don't, accumulator, non-accumulator. This compound has three-dimensionality that is in line with this globularity parameter. This compound does not. And this compound has the rigidity, rotatable bonds less than 5, and accumulates, and this was not. And we've done this for a bunch of examples. We spent a long time trying to understand what these mean, and the way these compounds get into bacteria. These gram-negative bacteria have porin proteins, so proteins that they use to take in nutrients. Okay, and these compounds co-opt this natural system. And this porin is an hourglass shape, and it has what's called a constriction region that's lined with negatively charged amino acids. And so us and now many others have done computational simulations to show that this amine positive charge interacts in a very productive way with the negative charge to facilitate the transport of this compound through. My former student, Sarah Perlmutter, asked the question about other types of positive charge. So again, as a chemist, we know that we can have a positive charge from an amine, but also from functional groups like a guanidinium and a pyridinium. And we know, and so what she did was assessed head-to-head compounds with each one of these and made the determination that yes, amines and guanidiniums are quite good, coefficient at this, whereas pyridiniums are not. My former student, Dr. Brian Drown, who's now a professor at Purdue, built a web app to predict accumulation. Okay, and I like to say it's so easy, even I can use it. Okay, so all you do is you take your ChemDraw structure and right-click on it, and you paste the SMILE string in, and this web app will compare the results to the entry rule cutoffs and will predict. So this compound meets all 3 entry rule criteria and would be predicted to be an accumulator, and this one does not and is not. And so this has been very useful to the community. There's been over 1 million compounds processed by people all over the world through Entryway. Okay, so of course what we want to do is make antibiotics, and so having this fundamental understanding now enabled us to go back and look at these promising compounds. So I mentioned all these before. And so all of these would be active, as mentioned. So we took these and applied the entry rules. And so I'll just talk briefly about these and then go into this one. And so Andrew first did this one. This is 6-DNM. This compound meets 2 of the 3 entry rule criteria. We now make a version that meets all 3. Okay, now this compound accumulates and now it kills gram-negative bacteria. Same story with ribacil C. My postdoc Steve Motika made this compound, now kills gram-negative bacteria. And then I think the example that's made it the farthest is this fatty acid biosynthesis inhibitor, so called WO1452. It meets 2 of the 3 criteria. We first made this simple addition, and you know, again, the whole idea of the entry rules is you can just within a few compounds make a compound that can be active. We've since optimized this to this compound that we call fabomycin, and this is advancing in partnership with different groups. The nice thing about this drug is this is a new antibacterial target. Okay, so fatty acid biosynthesis in bacteria is very different than in humans, so makes it a good target. And as I mentioned, we also think it's a good target to kill pathogens over commensal bacteria. So there's an enzyme that takes this double bond to a single bond called FABI, and it's different enzymes called different things in different organisms. This had been discovered at GSK as an inhibitor of this enzyme. It traded hand was optimized into this compound that I showed you before, super, super potent against gram-positive bacteria, no activity against gram-negatives. This is in phase 2 for staph infections. And again, one of the provocative things for us was that the pathogenic bacteria, staph, E. coli, Klebsiella, abamani, possess FABI. A few others do not, but most importantly, human commensals have this different version, FABK, or a very different version of FABI. So we thought there was potential to kill the pathogenic bacteria, the infectious bacteria, and not the commensals. And when we started this, there's hundreds of derivatives out there, and none of them had notable gram-negative activity. So my postdoc at the time, Dr. Erica Parker, looked at this and saw that we had 2 of the 3 criteria. I showed this before, super potent, but the potential is there. And so what she did was looked at there's an X-ray structure, or a structure of the compound bound to FABI, okay? And she noted a region here that didn't seem to interact with the protein. So maybe we could put a functional group on that could help the compound to go into the bacteria. And so again, she made just a handful of molecules where we put an amine at this position, and molecular docking suggests that this indeed points out into the solvent, so away from the protein. This compound, we call W1452 amine, meets all the entry rule criteria, as opposed to this one and this control compound. This compound now accumulates in gram-negative bacteria. It remains potent against staph, and now picks up activity against these gram-negative pathogens. Pseudomonas has a different set of FABI, so it's not expected to be active. In this field, you understand how the compound kills by generating resistant mutants. So again, you have 1 billion bacteria in a flask, and you can treat. And one thing that we noted, it's very hard to generate resistance to this compound, which is good. But when we do generate resistance, it's a mutation in this FABI protein, okay? And from the X-ray structure, we know that's very close to where the compound binds. We do mouse infection models, and so one of the nice things with antibiotics is you can infect a mouse with a bacteria, and it's actually fairly similar to a human infection, okay? And so we use this abamani, so these are the so-called escape pathogens, and so the parental compound, these mice succumb to the infection, and we save a large fraction of them with W1452 amine. Again, colony-forming units, or amount of bacteria in the lung after the infection, is way down when treating this. So in this first paper, we showed this was active in a bunch of different mouse models. But of course, we wanted this compound to be better. And in medicinal chemistry, what one does is there's effectively an infinite number of different compounds that one can make. And this is both a blessing and a curse, right? Like, again, there's always a better compound out there. And so we look for different regions of this molecule that we can alter to improve activity or reduce side effects, et cetera. And this was a collaboration with Professor Deb Hung at the Broad and Erica and Brett Kane in my lab. And so what we did was we made a series of molecules, okay, and we test them against these drug-resistant gram-negative pathogens. And so there's a bunch of compounds here. And we hit on a compound, compound 7 here, and this is a mixture of 2 non-superimposable images, 2 enantiomers, and all of the activity resides in this enantiomer, and we call this compound fabomycin. So fabomycin, we've partnered with Walter Reed they have a panel of 100 pathogenic bacteria that they isolate contemporaneously from patients, and they test fabomycin against it. And so this is compared to a fluoroquinolone, levofloxacin. And the thing you can see here is some bacteria are very sensitive to levofloxacin, but many bacteria are quite resistant, whereas fabomycin kills them in a very narrow concentration range. And this is with abamani, and it's a very similar thing with Klebsiella. So a group of bacteria that are not sensitive to levofloxacin, but are sensitive to fabomycin. Excitingly, when we tested this against gut commensal bacteria, we saw that it does not kill any of them, okay? So this was just in culture, where we can grow these gut commensal bacteria, and doesn't represent the whole diversity of the microbiome, okay? And I'll get to that, but this was an exciting start. We've learned a lot about fabomycin, so we did get an X-ray structure of it bound. So this is the compound bound to the protein, and we understand it to have this low frequency of resistance because it tends to interact with the backbone of the protein and not the side chain amino acids that become mutated. Fabomycin is also quite efficacious in rodent models. So this is a sepsis model in mice, fabomycin and the vehicle, a sepsis model in rats, again, fabomycin and the vehicle. And then this led us to do a UTI model. And so the UTI model is really a proving ground. Typically when drugs are evaluated in people, the UTI is the first place that it happens because those tend to be, in some cases, very drug resistant and difficult to treat. And so we partnered with the NIAID to do this, and they assessed fabomycin in different dosing schedules. They used colistin as the control. Colistin is an interesting antibiotic. It was, in the 80s, it was taken off the market because about 50% of patients experience acute kidney toxicity from colistin, but it's just been reintroduced recently because of the clinical need. And so when we look at bacteria burden in the different organs after treatment with fabomycin, we can see that it compares quite favorably to colistin in most of these evaluations. Okay, so this has been exciting, and more broadly, the actionability of the entry rules has been great to see. So we put these rules out there in 2017, and now many different groups have used them. There's this drug from Genentech, this one from Roche, this is from a group in Oxford, this is from Merck, this is from a group in Denmark. All of these are advancing as antibiotics. All of these were developed using the entry rules, and there's probably a couple dozen other compounds in the literature that have been developed using the entry rules. So we're very, very excited about that and proud of that, and glad that others have found these rules to be useful. We've turned our attention over the last few years to this other organism. So Pseudomonas aeruginosa is a very different pathogen to E. coli, which we developed entry rules for. And we found that the entry rules don't really apply to Pseudomonas. So there's some compounds that will accumulate in Pseudomonas that meet the entry rule criteria, but others do not. And this was not so surprising. E. coli has these general porins that I talked about, whereas Pseudomonas has maybe 40 specific porins. And so we redid this whole experiment, just trying to understand compound accumulation in Pseudomonas, okay? And I give a lot of credit to my former student, Emily, my current student, Morgan, for taking this on, because it looked perhaps like we could not find general rules for Pseudomonas. So we expanded the compound collection for this. We used our compounds, but also partnered with Roche, where we got access to about 20,000 of their compounds. We selected 150. We did this whole experiment again, where we assessed compounds one at a time. And with machine learning, we developed what we call the passage guidelines, Pseudomonas aeruginosa self-promoted entry. And the passage guidelines stipulate these parameters. If it has a certain formal charge or hydrogen bond donor properties, those compounds will accumulate and kill the bacteria. Okay, so this is a summary of what we've done at this point. So the entry rules are for E. coli. The passage rules are for Pseudomonas. All right, for this last part, I just want to turn the attention back to this point, okay, that I made, and just really more purposefully thinking about trying to develop antibiotics that will kill pathogenic bacteria, but not normal gut bacteria. And so this is what's happening now. The antibiotics we take kill the target infection, but they also kill off-target microbiota. So these are both gram-negative and gram-positive organisms. And then one gets dysbiosis. And so one can see this. People have studied this. So you take antibiotic, very simple experiment. You grow these commensal bacteria, and you look at different antibiotics. These are papers from others, and basically showing that all these antibiotics will kill these commensal organisms. As I mentioned, this is having really devastating consequences. And so we know that patients with colorectal cancer have a much less diverse microbiome. And so the cause and effect here is not totally clear, but it does seem like this promotion of colorectal cancer and irritable bowel disease is coming to the fore. As I mentioned, associated with depressive syndromes, motivation for exercise, and then the big one is C. diff infection. So almost 500,000 of these every year, 29,000 deaths, lots and lots of complications within the hospital. And again, this is because once taking these antibiotics and just wiping out your gut commensal bacteria, these bacteria regrow disproportionately and cause this infection where there's very few drugs to treat it. People actually give fecal microbiome transplant. That's one of the ways that we treat this. And I'm hoping we'll look back at this as like the medieval times for treating C. diff, right? And wouldn't it be great to not kill those bacteria in the first place? All right, so this is the question. How do we discover antibiotics that kill these pathogenic bacteria but not gut commensals? Very challenging. Bacteria are evolutionarily related. A lot of the same machinery for taking in and processing compounds and metabolites. What we focus on is the periplasm in gram-negative bacteria. So the periplasm is the space between, I told you gram-negative bacteria have this extracell wall. The space between the inner membrane and outer membrane is called the periplasm. It's only 7% of the cell volume, okay? But if you hit a target in the periplasm, by definition, it doesn't kill gram-positive bacteria, right? Gram-positive do not have the periplasm, so that's good. But also, a lot of these targets in our analysis, they tend to lack homology with the same targets in commensals, meaning the protein is not similar to the protein in commensal bacteria. Periplasm is if you have a compound that concentrates, you really need a vanishingly small amount of it. 2,000 molecules will get you a concentration of 1 micromolar. So maybe this accumulation problem is not as acute. And so this is our question. Can we develop a periplasmic targeted compound that can be pathogen selective antibiotic? Okay, so we focused on, doubly selective, I mean selective for gram-negative or gram-positive, and then selective for pathogens over commensals. And so the periplasm has these different machinery. This one that we focus on for processing lipoproteins. And the thing that we like about this is it's exclusive for gram-negatives. It's really important for the cell. It's conserved across gram-negative pathogens, and gram-positives don't have it. And so this is the pathogens. So if you look at these LOL proteins in E. coli, Klebsiella, Enterobacter, these are key gram-negative pathogens. They're very similar. And then if you look at it in the commensals, now they're different. Okay, so the idea is like, maybe we can come up with a drug that interacts with this version, okay, but not with this version. Okay, so this was our goal. We wanted to target this complex. We were able to really learn from the literature. AstraZeneca had published a few nice papers about compounds that hit this pathway, okay, these two compounds. These compounds had some activity against E. coli. They had no activity against these other gram-negative pathogens. And they know activity, of course, against gram-positive, but we thought that there was a glimmer of activity and a glimmer of hope here. So my former student, Dr. Kristen Munoz, took this on. We took these two scaffolds and on paper, merged them and did structure-activity relationships. So made approximately 50 compounds, Kristen made, and we came upon this compound, lolamycin. Okay, lolamycin is now active where the progenitor compound is not against E. coli. It picks up activity against these other gram-negatives. But of course, it doesn't have activity against Pseudomonas and Acinetobacter. These are very different, or staph. This is gram-positive. But what we want to know is its activity against these human commensals, which are not similar. And so we were really delighted to see in these human commensals, again, grown in culture, this compound has no activity. It also has no activity against gram-positives. Of course, this is just in culture, and so we need to do more experiments, as I'll discuss. Okay, so we first showed that when we generate resistant mutants, you have a mutation in this LOL gene consistent with it hitting its target. And we know from three-dimensional studies and computational modeling that the compound binds to this LOLCE interface. Okay, as a prelude to the microbiome study, we wanted to see, is this compound effective as an antibiotic? So again, we can show that this compound reduces bacterial burden, okay, by orders of magnitude, and it spares mice. Okay, so this is mice affected with different bacteria and their activity with lolomycin and the progenitor compound. We also know that it's effective given orally, so this is injection or oral. The compound has oral bioavailability about 50%, which is quite good. It's well tolerated in these animals, so we can give very high doses, and it's effective when given orally. Okay, so this sets us up for the big microbiome experiment. So the gut microbiome in mice is really a very good mimic for that in humans. And so we can ask this question, what effect does lolomycin have on the microbiome in mice? And so this is how we set this up. We have 4 groups: vehicle, amoxicillin, a broad-spectrum antibiotic, clindamycin, gram-positive only, and then our novel antibiotics, lolomycin, gram-negative only, and that we think is gut microbiome sparing. And so we get these mice, they acclimate, we treat according to the therapeutic level of these, and then we start collecting fecal samples day 0 before treatment and collect them all the way out to day 31. And again, I mentioned now modern DNA sequencing, you can extract DNA from these fecal samples, sequence, and you can know the distribution of all the bacteria. It's really, really remarkable, very, very positive technology. Okay, so this is just bacterial abundance, and there's a first question, like how many bacteria are left after treatment? Okay, and so this is a heat map where color means there's more bacteria. This is a group treated with vehicle. This is a group treated with lolomycin, so you can see all these bacteria. And then you can, the first thing that you see is like clindamycin at day 7 and amoxicillin at day 7, just like a wipeout. Okay, very, very few bacteria left at all. Then you can look at in the bacteria that are left, what do they look like? What are those bacteria, right? And so this is these composition plots, and these are the different family, at the family level, bacteria. And so vehicle, day 0, day 7, day 10, day 31, amoxicillin, clindamycin, you see a wipeout and then a takeover by certain bacteria, right? Consistent with what we know. Certain bacteria will regrow disproportionately, and whereas lolomycin does not have that effect, so no disruption at the family level. Another thing we do is look at the diversity. So this is the diversity, the range of different intercellular organs or microorganisms. So again, this is vehicle, day 0 and day 7, lolomycin, day 0 and day 7, and then just look at this. So this is amoxicillin, and then way down, and clindamycin, day 0, day 7, way down. This is species richness, which is the number of different species in a community. So beforehand, and then boom, day 7, way down, day 31, this is still not recovered. Okay, so the final experiment is the C. diff challenge experiment. So the idea is if we treat with antibiotic, then challenge with C. diff, will that C. diff infection take hold if the gut microbiome is wiped out, or will it not if it's maintained? So how does this treatment affect the clearance of C. diff? And so the same experiment, same group of animals, treatment, but now we challenge with C. diff, and then you can collect fecal samples and culture on a C. diff specific media and count those colonies. So again, vehicle, after treatment and challenge with C. diff, collect fecal samples, you see very little C. diff. This is a log scale. Amoxicillin, big take in C. diff. Clindamycin, again, this is a log scale, so orders of magnitude high. And then with lolomycin, very low on this. So really excited about this as a means to specifically target these pathogenic bacteria and not wipe out the gut commensals. Okay, so that's what I wanted to tell you. So we've been focused on really this process using entry rules and passage rules to take antibiotics of interest and make them into ones that are active against gram-negatives, but others are doing other things. So for example, Professor Laura Pittick has basically made a screening deck that meets the entry rules. And so instead of having 8 million random compounds, 50,000 compounds that now comply to the entry rules, and she was able to find hits with that relatively small collection. Whereas Professor Seiber at University in Munich synthesized a biased collection of compounds using entry rules, and again was able to identify hits. And so these fundamentals of compound accumulation led us to these two molecules. As I mentioned, the university has licensed these, and I'm really excited for these to move forward and see if they can help and hopefully be on a new wave of antibiotics that is better for patients and is microbiome sparing. More generally, we really are taken with this workflow, okay, where we have a collection and we measure accumulation and then this artificial intelligence and machine learning and applying lessons. And so my student, Aya Kelly, Dr. Aya Kelly, my former student, did the same thing with the blood-brain barrier. And so she developed a cell-based assay for that, identified traits for compounds that allow it to evade the blood-brain barrier and get in, so to treat neurodegeneration or brain cancers. And then she took this drug that has little penetrance in the brain and made it into an anti-cancer drug that does, and showed that it had enhanced survival in a mouse model. So this process is one that we're really excited about. That's what I wanted to say. I'm really grateful. I pointed out my students along the way, amazing group, current group and former group of students. Grateful to my collaborators at the University of Illinois, but also at Walter Reed and Roche and the Broad Institute, to the NIH for funding. And thank you again for the invitation. I'm really happy to be here and happy to take any questions. Thank you.